Very good morning. So, today we are going to learn the chapter photosynthesis in higher plants. Like how is we have learned nutrition in class 10th, right? Nutrition in plants we have learned. Connecting that chapter, linking with photosynthesis in higher plants. And whatever content we are going to learn, on that we are going to solve previous year questions as well. So, yes, now let us begin. First with the discoveries of photosynthesis. Discoveries of photosynthesis, which in your NCERT or at most 2 pages content is there. From that, what questions you will get and in what way you can learn it from a single slide, I will show you now. So, yes, tell me the standard equation of photosynthesis before. 6 CO2 plus 12 H2O in the presence of chlorophyll and sunlight. C6 H12O6 plus 6 H2O and oxygen is liberated as a byproduct. The standard equation we all know, right? Now, discoveries, what discoveries of photosynthesis will tell you? These are the raw materials of photosynthesis. This is the primary product of photosynthesis. This is the byproduct of photosynthesis, were all discovered in a sequential flow. First tell me, who is the scientist who said that carbon dioxide is essential for the process of photosynthesis? Who said that, beta? Carbon dioxide is essential for the process of photosynthesis. It is from Mole's half-leaf experiment. Yes or no? Mole's half-leaf experiment. We all are aware of this experiment from our 10th standard, right? So, need not to have a detailed discussion on it. Next, who is the scientist who said that water and in the presence of chlorophyll molecule, in sunlight and water, photosynthesis is conducted and oxygen is evolved from photosynthesis. These points are delivered by which scientist? Very good, Jan Ingenhousz. Okay, Jan Ingenhousz said that water and sunlight are essential for photosynthesis and oxygen is liberated from photosynthesis. What is the model organism he took? Hydrilla plant, which is an aquatic plant, right? Next, who said that glucose is the primary product of photosynthesis? Very good, Von Sachs. By conducting what? Starch test to the leaves which have bell pepper on it, okay, where in half of the leaf, some part of the leaf which is not exposed to sunlight is turning to pale color after starch test. The part of the leaf which is exposed to sunlight is turning to blue black color. Next, so from this we are aware like carbon dioxide is essential for photosynthesis done by Mole's half-leaf experiment. Water, sunlight are essential and oxygen is evolved from photosynthesis is said by Jan Ingenhousz. Glucose is the primary product, oxygen is the byproduct of photosynthesis. Now, you tell me who discovered oxygen? Oxygen discovery by Joseph Priestley. Joseph Priestley. Okay, oxygen discovery is done by whom? Joseph Priestley. And oxygen is evolving out from the photosynthesis is said by Jan Ingenhousz. Whether it is from carbon dioxide or from water is a big question for the scientists. Okay. So, oxygen is coming from water, not from carbon dioxide is said by which scientist? C.B. Van Niel. It is said by C.B. Van Niel. He said oxygen which is evolving out of the photosynthesis is coming from water, not from carbon dioxide. What type of photosynthesis did he conduct and gave this output? He has conducted bacterial photosynthesis. Okay. C.B. Van Niel has conducted what photosynthesis, beta? Bacterial photosynthesis. Bacterial photosynthesis, where you can also call it as anoxygenic photosynthesis. In this, the electron donor will not be water. What will be the electron donor? H2S. H2S is the electron donor, and if we can see the products out of after the photosynthesis, free sulfur molecules are present. When this is the case, in oxygenic photosynthesis, oxygen is coming from Water. So, are you all clear with the discoveries of photosynthesis? Who said that carbon dioxide is essential for photosynthesis? Moles, half-leaf experiment. Water and sunlight are essential and oxygen is evolved as byproduct is said by whom? Jan Ingenhousz. What is who said the primary product of photosynthesis is glucose? Von Sachs. And who discovered oxygen? By what experiment? We are very much aware of it from the schooling, right? Bell jar experiment. Okay, what is the experiment beta? Bell jar experiment. And C.B. Vanil has conducted what photosynthesis? Bacterial photosynthesis, and said that oxygen is coming from water. Many a times from this discoveries part, questions are asked in the match the following type. Okay. So, just do not neglect this, this is highly useful, and learning all the discoveries in this framework will just finish your revision in just 2 minutes. Okay. I will save this bit Aisha, okay, you can note it at that time. Now, one more scientist who said like sunlight is essential for photosynthesis, of course, Jan Ingenhousz has told. What spectra of sunlight is useful? What spectra of sunlight is in that spectra of sunlight photosynthesis is maximum. Who said that? It is said by E.W. Engelmann. Who said it, ma? E.W. Engelmann. What did he do? Okay, he gave us both action spectra and absorption spectra. Okay, he gave us both action spectra and absorption spectra. He has conducted algal photosynthesis. How? Okay, first he took a prism and gave a unilateral sunlight to the prism, and then after projecting it to prism, how will it scatter beta? It will scatter into VIBGYOR. Okay, it will scatter into VIBGYOR. Okay. This scattered light is being exposed to algae. What is that algae? Cladophora. Cladophora, which is a filamentous algae. So, this light is exposed to what? Filamentous algae. Over this algae, bacteria were placed. What are those bacteria? Aerobic bacteria. Listen to this carefully. Okay. So, what is the setup beta? Unilateral sunlight exposed to prism, then scattered to VIBGYOR. This light is being exposed to Cladophora, which is a green filamentous algae. Over this Cladophora, what is there? Aerobic bacteria. So, why aerobic bacteria is placed? Just now we have learned that oxygen is evolved as a byproduct. Wherever there is maximum oxygen evolution, there you can see peaks of aerobic bacteria occupied. Okay. So, like that when you check the maximum amount of photosynthesis, it is very clear, you can see maximum amount of photosynthesis at blue and red light regions. Yes or no? At blue and red light regions, maximum photosynthesis is conducted. So, yes, with this he said that at blue and red regions maximum photosynthesis is conducted, and thereby he proposed action spectra and absorption spectra. From this only we got to know the colors of pigments and what are the range of pigments that we have also. So, what is absorption spectra? Absorption spectra is a graph wherein you can see different pigments absorbing light at different wavelengths in photosynthesis. What is action spectra? Maximum amount of wavelength or maximum amount of photosynthesis conducted by the pigments is projected in that graph. So, now if you see both absorption spectra and action spectra of your NCERT, what you can see first in absorption spectra, you can see the core pigment and antenna pigments also, chlorophyll A, chlorophyll B, carotenoids. Colors of the pigments are very important beta, where we got to know this from the chromatogram. Chlorophyll A is blue green color. Okay. Chlorophyll A is of what color ma? Blue green color. Chlorophyll B is yellow green color. Carotenoids are yellow orange color. Okay, yellow orange color. Xanthophyll is also antenna pigments or accessory pigments, it is of yellow color. Okay, clear with the colors? Chlorophyll A is of what color? Blue green. Chlorophyll B? Yellow green. Carotenoids? Yellow to orange. Xanthophyll? Orange. Okay. Of this you remember, the pure accessory or antenna molecules or pigments are nothing but they will absorb light at different wavelengths, okay, like 400 nanometers to 500 nanometers, carotenoids and xanthophylls. Chlorophyll B also like that only, like it is not the core pigment, maximum wavelength it is not absorbing. Chlorophyll A is absorbing maximum light, okay. If you can clearly see this absorption spectra with different pigments, chlorophyll A, if you have to see, you can see one peak in between 400 to 500 nanometers, yes, and one more peak in between 600 to 700 nanometers. If you have to check the same for chlorophyll B, you can see only one highest peak in between 400 to 500 nanometers, and this is small peak, we are not counting it. Next, carotenoids, you can see two peaks only between 400 to 500. When this is the case, from this you can clearly state now which is the pigment that is absorbing the wavelength maximum. Chlorophyll A is absorbing maximum sunlight. Very good morning Anisha, just go through the class beta, this is highly useful for you. Next, very good that you have predicted Engelsmann experiment and red and blue light also, yes. If you have to see action spectra, okay, in action spectra you can see two peaks, okay, one in between 400 to 500 nanometers and other is in between 600 to 700 nanometers. Focus here, be here. This, is it not same as that of chlorophyll A from the absorption spectra? Yes or no? So, here there is a clear statement that chlorophyll A is the chief pigment and chlorophyll A of the absorption spectra is same as that of action spectra. Okay. So, here is the graph of chlorophyll A, how we can synchronize that chlorophyll A of absorption spectra and action spectra. Fine, got it? Clear with absorption spectra and action spectra? Core pigments, fine. E.W. Engelmann's experiment, fine. All the other experiments also, good. Now, firstly we have to go through the chloroplast structure and then we will go to the reactions in the photosynthesis. What is the site or the organelle where the photosynthesis is conducted? What is the organelle beta? Inside the plant cell. If I have to suppose this is the plant cell and you got a question, giving 4 options, you got a question, which cell will conduct photosynthesis, and you were given with the options mesophyll, chloroplast, chlorophyll, what you will choose, or all of the above. Which cell will conduct photosynthesis? Site of the reaction is always important. In biology, do remember ma, site of the reaction or site of the function is very much important. Look into like what is he asking in the question, is he asking about cell, is he asking about cell organelle, or is he asking about the pigment, okay. So, if he is asking you about the cell where the photosynthesis is conducted, what should be your answer? Mesophyll cell. Which organelle? Chloroplast organelle, okay. Inside chloroplast, where is the absorption of light taking place? The thylakoids, this I will tell you, okay. Cell and cell organelle is clear to you know. Now, looking into the cell organelle. It is a single membrane bound or double membrane bound organelle? Double membrane bound organelle, and it has membranous sac like structures. These are the membranous sac like structures. What do you call it as? Thylakoids. Okay, membranous sac like structures called as thylakoids. This membrane is called as thylakoid membrane or thylakoid lamellae. Fine, and at the center this is thylakoid lumen. Fine, this is thylakoid lumen and this is membrane is thylakoid lamellae. Stalk of this sac like structures, what do you call it as? Grana. Stalk of the sac like structure is grana. One grana is connected to other grana by what? Stroma lamellae. One grana connected to other grana by stroma lamellae. And inside the stroma, if you can see, you can clearly look into starch granules and lipid granules. whichever diagram is there in your NCERT, fix the labelings in your mind. Okay. What type of ribosomes can you see beta in chloroplast? Nandu, you tell me, Nana, is it 70S or 80S? 70S ribosomes. If it is 70S ribosomes, what will be the larger subunit of it and smaller subunit of it? 50S and 30S. Very good. 50S and 30S. What is the main activity of ribosomes here? Protein synthesis. And what protein is it synthesizing inside the chloroplast? Remember, Gurdechko. World's abundant protein, Rubisco. Okay, it is synthesizing world's abundant protein, that is Rubisco. Okay, so it has 70S ribosomes, whose activity is protein synthesis, and it is synthesizing world's abundant protein. And there is one more thing we have to recall with respect to chloroplast, that is, it is having self DNA, that is what? Single stranded, double stranded circular DNA. It has what, Nana? Double stranded circular DNA. This double stranded circular DNA also. What is the ultimate goal of DNA? Protein synthesis, that is Rubisco. Fine. Finally, you have to remember Rubisco is being synthesized and it is having its own DNA, that is double stranded circular DNA. That is why chloroplast is called as semi-autonomous organelle or self-duplicating organelle. Fine. Is that clear with the organelle chloroplast? Everything is fine, right? Now, so here I need to add on a slide. Now look at here properly, ma. Photosynthesis, whenever we have to remember, will get immediately to our mind is light reaction and dark reaction, of course, right. So here we have two reactions which are nothing but light dependent and light independent reactions. Shortly we will call it as light reaction and dark reaction. So here if you see light dependent reactions, light independent reactions. Under light dependent reactions, three set of reactions takes place. First is what? Absorption of light in the form of photons. The first thing is what? Absorption of light in the form of photons. Second is what? Photolysis of water. What is the second thing, beta? Photolysis of water. Water gets photolysed to 2 protons plus 2 electrons and oxygen atom. Okay. Third is what? Formation of assimilatory power. Formation of assimilatory power. What is the assimilatory power over here? ATP and NADPH. Fine. So these are the 3 steps of photosynthesis. I already told you, locations are important again. Where is the absorption of light in the form of photons taking place? Online students also answer, beta. Absorption of light in the form of photons takes place in the which location of chloroplast? Very good, Akshita. It is thylakoid lamellae. Photolysis of water takes place, Nandu, lumen. Okay, that is inner membrane of thylakoid towards lumen. Inner membrane of thylakoid towards lumen. Formation of assimilatory power takes place where? No, it takes place in formation of ATP and NADPH in stroma. Clear kada locations? Fixed in your mind? Yes. Now, when it comes to light independent reactions, okay, light independent reactions. Light independent reactions means what? Carbon dioxide is fixed to glucose. Keep it in your mind, carbon dioxide is reduced to glucose or carbon dioxide is fixed to glucose is one and the same. And what is the primary product of photosynthesis? Glucose only. Ultimately, glucose should be formed, right? So, yes, carbon dioxide is fixed to glucose. Done? Or reduced to glucose. Okay? So, here it requires ATP and NADPH for this process. Okay? So, the assimilatory power whichever is formed at the end of light reaction is utilized for dark reaction. Okay? Calling light independent reaction as dark reaction is a misnomer. Remember this statement. Why are we calling it as misnomer? Because dark reactions does not mean it takes place in dark only. It is independent of light. It is dependent on the products of light. Whichever statements I am using, right, listen it carefully. So, this reduction takes place in different pathways like C3 cycle, C4 cycle and CAM cycle in different plants. Okay, in different plants it is of different cycles, but utilization of energy by all these 3 cycles, that point is common, and level of energy utilized is also different, time of utilization is also different, what are the cells which are involved in fixation are also different with respect to these 3 cycles. Now, we will go and look into the sequential steps of light reaction and then dark reaction in brief. Okay. So, now if you have to see again, I told you first is what? Absorption of light in the form of photons. Consider this as only thylakoid. Okay. From this, let us suppose this is the chloroplast cell. This is the chloroplast, let us suppose. Okay. Or else I will draw double membrane bound organelle only. This is a chloroplast, let us suppose, and inside that you have thylakoids. Okay. One thylakoid I have drawn here. Okay. This is one thylakoid. This is the membrane and this is lumen. In thylakoid membrane, you will be having what? Photosystems. What do you have? Photosystems, PS2 and PS1, photosystem 2 and photosystem 1. How photosystem 2 and photosystem 1 were placed? Based on their discoveries, that is it. Okay. Photosystem 2 and photosystem 1. Inside this photosystem, what do you have is light harvesting complexes. Nandu, recall everything beta. Light harvesting complexes. Light harvesting complex has both accessory pigments and one more is what? Core pigments. What are the accessory pigments? Chlorophyll, B, carotenoids, xanthophylls. These are the accessory pigments. Chlorophyll A, carotenoids and xanthophylls are the accessory pigments. Like inside the photosystem 1 or photosystem 2, these will be the accessory pigments. They will absorb light at different wavelengths and they will submit it to what? Core pigment. Core pigment is what? Chlorophyll A. All these pigments are present where? Inside photosystem, inside thylakoid lamella itself. Is that clear? Okay. So, first thing is what? Absorption of light in the form of photons. That thing is clear to your mind. Absorbing the light, the water whichever is present inside the lumen of the thylakoid, this water gets splitted to 2H plus 2 electrons plus oxygen atom. Okay. These 2 electrons whichever are formed over here in the lumen, they get excited and they activate the process that is non-cyclic photophosphorylation in green plants. Okay. What do they activate, ma? Non-cyclic photophosphorylation. The benefit of biology is the process is in the name itself. Okay. It is non-cyclic photophosphorylation. Non-cyclic means it is not in a cyclic pattern. Photo means light, phosphorylation means addition of phosphate group. Okay, utilizing light, addition of phosphate group, that is non-cyclic photophosphorylation. Where does this happen? In thylakoid lamellae itself or thylakoid lumen itself, here in the photosystems. Okay. Electrons whichever are formed after photolysis of water, they are getting excited. So, now these 2 electrons will get excited and they are given to what, Nana? Core pigment. Okay. They are given to what? Core pigment, that is chlorophyll A. Okay. And the maximum absorption peak of photosystem 2 is what? 680 nanometers. Okay. Chlorophyll A gives the first electron acceptor from here is pheophytin. What is the first electron acceptor? Pheophytin. First electron acceptor from here is what? Pheophytin. From pheophytin it is given to what? plastoquinone. Okay, plastoquinone, which is accepting 2 electrons from phaeophytin in thylakoid lamellae, parallelly consumes H plus from stroma. Okay, it consumes what? H plus from stroma, and this get converted to plastohydroquinone. These 2 H plus are submitted to what? Thylakoid lumen, and from here electrons are delivered to cytochrome B6F complex. Done. And the last electron acceptor of photosystem 2 is plastocyanin. Is that clear, ma? From here to here it is what? Photosystem 2. Fine. Now from plastocyanin directly again uphill movement is shown. Okay. Electrons are accepted by what? P700, highest wavelength. Okay. And here the primary electron acceptor will be ferredoxin. The next is ferrous sulfur complex. What are these protein molecules? Ferrous sulfur complex. And the ultimate electron acceptor from ferrous sulfur complex is located ferrous sulfur complex, and from ferrous sulfur complex 2 electrons are given to NADP plus. To what, Nana? NADP plus. Here I will write, it will be visible to you. This NADP plus is attached toward the membrane, towards the stroma. Okay? And utilizing 2 electrons, this forms out to be NADPH. Okay? And forming what? By the enzyme NADP reductase. Okay? So, what is the ultimate electron of the non-cyclic photophosphorylation electron acceptor? The ultimate electron acceptor of non-cyclic photophosphorylation is NADP plus, which is located attached to the membrane towards the stroma. Utilizing the electrons, it is getting converted to NADPH by the enzyme NADP reductase. Done? Fine, clear to you? So, here if you can clearly observe, this entire process takes place in the thylakoid lamellae itself in both the photosystems. This occurs in 2 photosystems. So, here if you can see in the photosystem 2, first electron acceptor is phaeophytin, last electron acceptor of photosystem 2 is plastocyanin, and primary electron acceptor of photosystem 1 is ferredoxin. Ultimate electron acceptor of non-cyclic photophosphorylation is NADP plus. Okay. So, this NADPH formation is happening where? Inside the stroma only. Clear in your mind? Fine. So, just at least if you are unable to remember the entire non-cyclic now itself, no issues, we will have some more classes on this, not at all a problem, this is a basic class only. At least remember the primary and ending electron acceptors. So, clearly if you can see beta uphill downhill movement of the electron flow, and this is the Z scheme, non-cyclic photophosphorylation Z scheme. Fine. We are not going to discuss cyclic photophosphorylation here, we are purely discussing oxygenic photosynthesis only. So, now you can see NADPH formation from the assimilatory power, which is the third step of light reaction. One more energy rich molecule is there to form what is that? ATP. Okay. ATP is, how ATP is formed is clearly explained by a hypothesis. What is that? Chemiosmotic hypothesis. Chemiosmotic hypothesis, which is given by whom? Peter Mitchell. This is given by Peter. Clear, Aisha? So, this how ATP is being formed is clearly explained through chemiosmotic hypothesis by Peter Mitchell. This concept is completely driven by one force, that is proton gradient. Proton gradient. Fine. And you have clearly understood the process of how the non-cyclic photophosphorylation is happening, right? There is a big paragraph of 3 steps in your NCERT where every student gets confused reading after that. No point of confusion, I will explain you all the 3 points clearly now itself. The very first thing is the primary electron acceptor, which is pheophytin, is delivering electrons to a proton acceptor, that is plastoquinone. Yes or no? Yes, it is giving electrons to a proton acceptor, that is plastoquinone. And this plastoquinone is accepting protons from stroma and it is giving to what, Nana? Thylakoid lumen. So, here is one place where because of plastohydroquinone, protons are getting reduced from stroma bank to, and profit is to lumen bank. Fine. From stroma, protons are submitted to lumen. Like that, proton reduction happened in the stroma. Second point is photolysis of water. Because of photolysis of water, protons increased in which bank? Thylakoid lumen. Okay. Second is protons increased in thylakoid lumen. Third point, okay. Ultimately, what is the ultimate or final electron acceptor of non-cyclic photophosphorylation? NADP plus, that we all know, right. So, this NADP plus is getting reduced to what? NADP H plus H plus. There also proton reduction is happening in stroma. Third is NADPH formation. Where, Nana? Inside stroma. So, because of these 3 points, okay, because of these 3 main reasons, the proton gradient is being started from stroma to lumen. First is what, Nana? You can consider photolysis of water as the first point. Second is protons being transferred from stroma to lumen by PQH2. Third is NADPH formation inside the stroma. Because of this, protons reduced inside the stroma, increased in lumen, thereby proton gradient is generated, okay, and this gives out ATP synthase formation. ATP synthase, whichever is there inside the thylakoid membrane. Again, this ATP synthase has 2 pumps, Nana, F0 and F1. F0 is embedded inside the thylakoid membrane, F1 is projected towards the stroma. In one pump, it generates 4 H plus in one turn, and in stroma you can see ADP and inorganic phosphate. This forms out to be ATP. Clear? Understood everybody? Okay. These are the 3 reasons of generating a proton gradient between stroma and thylakoid lumen. Because of the proton gradient, ATP synthase which is there in the thylakoid membrane is activated, F0 is embedded inside the membrane, F1 is towards the stroma. Like that, in a single pump it is generating 4 protons and forming ATP inside the stroma. Okay. Now, isn't it phosphorylated? Addition of phosphate group to ADP and ATP is being formed. This is how ATP and NADPH are formed inside the stroma. Now, utilizing this energy, we have to conduct the final step. What is that? Formation of primary product of photosynthesis, that is glucose, okay, utilizing this assimilatory power. So, now if you have to see that how it happens, I told you in 3 cycles, na beta, it occurs in 3 cycles. Just pay attention for 15 to 20 minutes, this will be clear to you. Okay. What are those 3 cycles? What is the only process that is happening over here? Yes or no? Okay. C3 cycle is activated in all the plants. Do remember, C3 cycle occurs in where beta? All plants. And C3 cycle is also called as Kelvin cycle, because after the scientist Melvin Kelvin, who has conducted algal photosynthesis and discovered C3 cycle. Okay. C4 cycle is observed only in the tropical plants. Okay. C3 cycle occurs in all the plants, and most of the plants who conduct only C3 cycle are temperate plants. clear na beta. And CAM cycle will be seen in the desert plants. Desert plants, or you can also call as Crassulacean family members. CAM cycle is observed in where? Crassulacean family members. Done? Easy, right? So, yes, only few points to remember. I will add on many other points when we are dealing with the advanced classes. So, C3 cycle is observed in all the plants, especially only C3 cycle is seen in temperate plants, C4 cycle is seen in tropical plants, CAM cycle, desert plants and Crassulacean family members. So, now if you have to see C3 cycle, why Kelvin has named it as C3 cycle? Because the primary product which is formed after carbon fixation is 3 carbon compound. Okay? So, here do remember beta, 1 carbon dioxide will enter the cell through what? Stomatal opening. So, stomatal opening will give the gaseous exchange, thereby carbon dioxide enters the cell. There should be one acceptor, no? Someone should invite when it enters the family, right? So, that invitation is done by RUBP for C3 family, C3 plants. Okay, in C3 cycle, what is the primary acceptor of carbon dioxide? RUBP. RUBP is a 5 carbon compound. Okay, and carbon dioxide is a 1 carbon compound. Okay, like that, in the presence of world's abundant protein, that is rubisco, carbon fixation or carboxylation, carbon reduction, one and the same. Okay. So, this carbon fixation takes place as, this is totally 6 carbons, right beta. So, 2 molecules of 3 carbon compound, that is phosphoglyceric acid. Okay, 2 molecules of phosphoglyceric acid are formed, which are 3 carbon compound. This is mediated by what? If at all in your question they are asking you what is the enzyme that is fixing carbon dioxide, your answer should be rubisco. What is the acceptor of carbon dioxide? Your answer should be RUBP in C3 cycle. Okay, in C3 cycle. Now, this 3-phosphoglyceric acid is reduced to 2 molecules of, here you can see, it is reduced to 2 molecules of glyceraldehyde 3-phosphate. Okay, 2 molecules of glyceraldehyde 3-phosphate. For this reduction step, okay, 1 carbon dioxide will utilize 2 ATP and it will utilize 2 NADPH. In a reaction, if the arrow is like this, so these are utilized. If something is over here, this you will see in the respiration, that means energy is released. Energy is released in respiration, utilized in photosynthetic cycles, like that you remember. Energy is utilized in photosynthetic cycles, released out in respiration cycles, like that you remember. So, here how many are being utilized beta for reduction process for 1 carbon dioxide? 2 ATP and 2 NADPH. From these 2 molecules of glyceraldehyde 3-phosphate, in 1 cycle, right, 1 carbon dioxide fixing means this is 1 cycle. 1 carbon will be escaped from every cycle. If 1 carbon is escaping from 2 molecules of G3PA, how many carbons will be remaining? 5 carbons. Those 5 carbons are regenerated back to RUBP. This regeneration requires one more ATP. Is that clear every one of you? Okay. 1 C3 cycle is clear to every one of you, right? First, what is the primary acceptor in the C3 cycle of carbon dioxide? Sit straight everyone, listen focusedly, it is RUBP. Okay, it is fixing how many molecules beta? 2 molecules of 3 carbon compound phosphoglyceric acid, and then it is reduced to 2 molecules of glyceraldehyde 3-phosphate by how much energy? 2 ATP and 2 NADPH. This is regenerating back, where 1 carbon is escaping to form sugar, 5 carbons are regenerating back to RUBP utilizing one more ATP. When 1 carbon from 1 cycle is escaping for the formation of glucose, how many carbons are required for the formation of glucose? 6 carbons. 6 carbons are required for the formation of glucose means how many cycles have to take place? 6 cycles. Okay, 6 cycles have to take place. In one cycle, if you have to count 2 plus 1, how much ATP? 3 ATP are utilized. One cycle 3 ATP, 6 cycles how many ATP? 18 ATP. Good. 6 cycles how many NADPH? One cycle 2 NADPH, 6 cycles 12 NADPH. Clear? And one cycle 1 carbon dioxide is being fixed. 6 cycles 6 carbon dioxide. So, for the formation of 1 glucose in C3 cycle, how much energy is required beta? 18 ATP, 12 NADPH. 6 carbon dioxides, 6 Kelvin cycles have to be processed to form a 1 glucose by C3 cycle. Clear? This takes place in what plants? All the plants. C3 cycle occurs in all plants. Only C3 cycle means temperate plants. And here there is one more point to remember you with respect to Rubisco, that is photorespiration, that I will deal in the advanced classes. Okay? Now, when it comes to C4 cycle, first you have to understand the anatomy of C4 cycle beta. Anatomy of C4 cycle means C4 cycle which occurs in the temperate plants, it has Kranz anatomy. It has Kranz anatomy. Okay, Kranz anatomy means the vascular bundles. What type of vascular bundles you will see in leaf, ma? Xylem, phloem. How they are arranged? What type of arrangement you call it as? Conjoint closed, right? Conjoint closed inside a leaf. Their xylem and phloem bundles are arranged how? Conjoint and closed. So, this vascular bundles are surrounded by or covered by bundle sheath cells. These bundle sheath cells will not have any intercellular spaces. You can see it over here. Is the image clear, ma? Can you see any intercellular spaces in between bundle sheath cells? No. They are arranged around the vascular bundles as a flower wreath. Okay? Flower wreath without any intercellular spaces, like that bundle sheath cells are arranged. Okay, with no intercellular spaces. And how the gaseous exchange takes place in between mesophyll cells and bundle sheath cells? By plasmodesmatal connections. What are the connections between mesophyll cells and bundle sheath cells? Plasmodesmatal connections. Okay. So, through plasmodesmatal connections, mesophyll cells and bundle sheath cells are connected. Bundle sheath cells will have agranal chloroplast. What chloroplast? Agranal chloroplast. Means they have chlorophyll, everything is there, but grana is not present, only stroma is there and rubisco is there inside the stroma. Okay? And mesophyll cells, they have as usual granal chloroplast. Clear with the Kranz anatomy of C4 plants, that are tropical plants. Few examples of C4 plants: sorghum, sugarcane, maize. Okay? All these are examples of what? C4 plants. And the Kranz anatomy is clear to you know. Around vascular bundles, what are surrounded? Bundle sheath cells, no intercellular spaces. How they are connected to mesophyll cells? By plasmodesmatal connections. Now, if you have to look into the C4 cycle, in brief I will tell you, in brief, and I will brief you the energetics also. So, if you have to see carbon dioxide, again it enters through stomatal openings. Once it is entering to the mesophyll cell, carbon dioxide is reduced to hydrogen carbonate by carbonic anhydrase enzyme. What is the enzyme? Carbonic anhydrase. This hydrogen carbonate is along with the primary acceptor of carbon dioxide. What is that? PEP, phosphoenol pyruvate. What is the primary acceptor? Phosphoenol pyruvate is the primary acceptor. This is fixing carbon dioxide in C4 plants. In C4 plants, what is the primary carbon dioxide acceptor? PEP, okay, phosphoenol pyruvate, that is 3 carbon compound. So, this is fixing it, carboxylation is happening, 1 carbon dioxide and 3 carbon plus 1 carbon, 4 carbon, right. So, oxaloacetic acid, which is 4 carbon, is formed. This is delivered to bundle sheath cells through what? Plasmodesmatal connections. And here this 4 carbon compound is decarboxylated to C3 acid and Carbon dioxide. Carbon dioxide will continue which cycle here? Kelvin cycle. Like that, let us assume for one carbon dioxide, okay, one carbon dioxide in Kelvin cycle which is happening inside the bundle sheath cells utilizes how much ATP beta? Very good, louder, be louder, 3 ATP and very good, 2 NADPH. Okay ma, okay, and this C3 acid whichever is there, that is phosphoenol pyruvate, is regenerated back. Okay. Here one more point to remember, at the time of fixation, okay, to oxaloacetic acid, this uses 1 ATP by C4 cycle. Okay. So, for one carbon dioxide fixation or one cycle, one complete cycle, it is using 3 plus 1 how much? 4 ATP and 2 NADPH, not 4 here, 2 ATP. 3 plus 2 how much? 5 ATP and 2 NADPH. But we all know 6 cycles need to be processed to form one glucose molecule. When 6 cycles have to be done, you calculate and tell me for 6 cycles, of course 6 carbon dioxides are required, and how much ATP are required for 6 cycles? Together 30 ATP. How much NADPH is required? Very good. Done. So, here what is the primary carbon dioxide acceptor in C4 cycle? Phosphoenol pyruvate. And what is the enzyme? Pepcase, phosphoenol pyruvate carboxylase. Okay. And C3 cycle is activated in bundle sheath cells. This for one carbon dioxide it utilizes this much of energy. Here for fixation of C4 cycle it utilizes 2 ATP. On total for 6 cycles this is the energy that is utilized by C4 cycle. Is that briefed into your mind? Okay. Now CAM cycle, C4 cycle, energetics are same beta. Like for one complete cycle it requires 5 ATP and 2 NADPH. For 6 cycles or for formation of one glucose it is same amount of energy, 30 ATP and 12 NADPH. But in CAM plants it is only one cell that is involved to conduct C3 and C4 cycle. Okay. In CAM plants what happens? In a single cell, that is in mesophyll cell, C3 and C4 cycles will be conducted. Okay. Wherein the carbon dioxide fixation happens during night time because of scotoactive stomata. What is scotoactive stomata? Any one of you? What is scotoactive stomata beta? The stomata which opens during night time, right? The stomata which opens during night time is what? Scotoactive stomata. That is an adaptation to prevent the process of transpiration by desert plants. Okay. So, this scotoactive stomata will open during night time and carbon dioxide fixation takes place during night time. So, C4 cycle, okay, C4 cycle will occur in the night time. Okay. And same cycle, the pepcase 4 carbon compound is formed, oxaloacetic acid, this is stored inside the vacuole. Where is it stored? Vacuole. 4 carbon compound is formed, that is stored inside the vacuole. During daytime this gets decarboxylation happens inside the mesophyll cells only, and C3 cycle is activated. Okay. C3 cycle is activated during what? Daytime. C4 cycle takes place in night time. Everything else is same. What is the primary carbon dioxide acceptor in CAM plants? Phosphoenol pyruvate, right? What is the enzyme? Pepcase. Clear? Same as that of C4 plants. Fine. Now C3 cycle, C4 cycle and CAM cycle is clear to you know. How glucose is formed is briefed to you know. Now based on the information that we have dealt till now, based on that only we are going to solve PYQs. It is just 50%. Whatever we have discussed today from photosynthesis is in higher plants, it is just 50%. Okay. But from that only we are able to solve the questions of all your PYQs. Just look into the most recently asked question, that is NEET 2025. Online students, you can also answer beta. Chlorophyll a color. Very good Akshita. Next. Option 4. Once again recheck Nandu. Chlorophyll B. Yellow green. Good. Good Nandu. Next. Xanthophyll. Yellow. Good. Carotenoids. Now you tell me the option beta. 4, is that correct? A3, B1, C2, D4. Done. Good. Good Nandu. Next. Very simple. 2024. Which of the following are required for dark reaction of photosynthesis? Dark reaction of photosynthesis. What are required? ATP, NADPH, ATP, NADPH and carbon dioxide. These are required for what reactions? Dark reactions of photosynthesis. So, what option? Good. Next. 2023. The reaction center in photosystem 2 has an absorption maxima at 680 nanometer. Very good. Next. How many ATP and NADPH2 are required for synthesis of one glucose molecule during Calvin cycle? As simple as it is. Next. Finally, statement based question from 2022. Online students, you also answer ma. So, first statement if you have to see, the primary carbon dioxide acceptor in C4 plants is phosphoenol pyruvate and is found in mesophyll cells. Correct only, right? Next. Statement 2. Mesophyll cells of C4 plants lack Rubisco enzyme. In the light of above statements, choose the correct answer, he is saying. Statement 2 also is right only, no? Mesophyll cells of C4 plants lack Rubisco. But bundle sheath cells of C4 plants will have Rubisco. Mesophyll cells will not have. Mesophyll cells will have what? Pepcase. Okay. What is the right answer? 1 and 2. Symbol ma, need not to confuse. In mesophyll cells, what do you have? Pep and pepcase of C4 plants. Bundle sheath cells, what do you have? Rubisco. That's it. Okay. If you are clear with those 2 points, you can answer this. Now, you have got some confidence about this chapter that you are able to answer few questions. We will learn this chapter in more detailed way once the program is starting. Okay? Thank you.